Okay, that was for all the people who paid attention to all of our methodologies for IVPT the entire day yesterday, the data analysis parts. So please join me in welcoming the panel for this session. Along with the three speakers this morning, joining us back on the panel, Dr. Theo Kapanadze, Dr. Lakshmi Raghavan, Dr. Sam Raney, Dr. Rong Wang, and also joining us on the panel is Dr. Abhishek Juluri. Dr. Juluri is an assessor within the Office of Bioequivalence, focusing on topical and transdermal products. Prior to coming to the FDA, he began his professional career as a senior scientist working in novel drug delivery technology for topical and transdermal delivery of macromolecules. And he also worked as a principal scientist at Targus Pharma, a topical CRO. So I think we have everybody on the panel. So in the interest of time, we are going to quickly get started. And we are going to start with questions from the room. Would anybody like to come up to the microphone?
Good morning. Thank you so much for the wonderful presentation. My question, how the receptor solution sampling qualification is assessed, considering it's an automated system and full replacement? Thank you.
Allison, Hiren?
Thank you for the question. So, I mean, the general answer is, as I mentioned in my presentation, there is a lot of evolvement in the equipment qualification. So I would highly encourage to utilize manufacturer specification to understand, and you have a good qualification data, and you are using the full replacement. So as long as you have sufficient data to say that every time using your methodology is a consistent volume, that should be sufficient. Thank you.
Thank you.
I appreciate the talk on statistical analysis. So just a scenario to where the validation study we run with the same quality management system as a pivotal. And the 4 to 6 donors is a recommendation. You could run more. If that data set demonstrated bioequivalence and the power analysis suggested that the number of donors was sufficient, is there a potential situation where the pilot could be submitted in lieu of the pivotal? And is there a suggestion based on your talk that if the pilot data is bioequivalent and the power analysis suggests maybe just a small number of donors, a pivotal study could be additive moving forward? Just your thoughts on that would be appreciated.
Thank you for the question. Let me say at this point that all our simulations and our work was based on a pilot study that did not consist of just 4 donors. So we consider cases where we had 6 donors or 10 donors in the pilot study. And as previously said, this is still at an exploratory stage. It definitely should not be understood as a part of the guidance. And if there is a suggestion, please talk to us and let us know. Also to point out that everything, a new method or everything that should be considered should be clarified and should be included in the study protocol in advance. And because we heard yesterday, we saw presentations about potentially including sample size re-estimation with outliers. We need to, in all cases, account for the potential inflation of type 1 error and adjust the significance level. But everything should be in advance included in the protocol and first discussed with us at the pre-under stage.
Thanks, Elena.
So my name is Kamlesh Amin and I'm from Solaris Pharma. So for method validation, the selectivity and sensitivity is the challenge for industry. And the industry need the guidance more in a detail. That's what individual variability is allowable to conclude the selectivity and sensitivity based on statistical model, right? That will be help to industry because we are looking individual replicate for the selectivity and sensitivity, but based on the skin nature is not the individual donor will be give the consistent data, right? So how much variability is allowed to conclude the selectivity and sensitivity, and what statistical model is good for that?
Sam, maybe you can start with what the expectations are, or Hiren, and then we can go to the statistical aspect.
So I don't want to set any number on the variability. I mean, first of all, thank you for the question. I think the challenge here is the lower number of donor for the sensitivity and selectivity and come up with a better quantitative tool to make a consistent decision, right? And as I mean, this is something, we are definitely, aware about the situation, and we will see, we are exploring, what can be done in the quantitative field. But as of our current practice is, we are looking at this data on individual donors, mean values per with the standard deviation and see. And I don't think so. The expectation is, if you are using 4 donors, all should show discrimination. As we see some of the data yesterday, it's just the nature of that particular donor where, there's a lot of uncertainty. So, I mean, yeah, I don't have a number, but, if majority of the donors shows discrimination in both sensitivity and selectivity, I think that should be sufficient. And yeah, I mean, yeah, I don't have any additional comments. You might.
Well, I think that's exactly right. The objective of the sensitivity and selectivity studies are really to guide us on what conclusions can we actually draw from the pivotal study. If the pivotal study appears to show that across the entire population of donors in the pivotal study, this is key, not in individual donors, across the population of the pivotal study, that the test and reference product are bioequivalent. Is that because the IVPT method can actually discriminate differences, but there were no differences, no significant differences? Or can we not conclude that? And so when we look at the sensitivity data, the first thing is look at the across donor population and maybe a 6-replicate pilot study, sorry, a 6-replicate sensitivity study, but look at the data, draw the conclusions about whether it's sensitive or not based across the 6 donors. And I think there, when we look qualitatively, we're looking for an ability, because the data set's a little bit too small to sometimes get a statistical conclusion. We are looking at other types of quantitative analyses. And, in time, we'll try to make those available once we kind of think they're ready to be reliable. But for now, it's a qualitative assessment to really say, just beyond the error bars, putting the error bars on those average across donor populations, are they separated? And they don't necessarily need to be separated at every single time point, but because of the early time points, they may not be separated. And at the late time points, they may not be separated. But especially if around J max, they're cleanly separated, I think that's compelling. There are other scenarios that can also be entertained, but I think those are the key things that I would try to convey, is that use the individual donor data primarily not to draw conclusions about sensitivity per se, but to understand, is my across donor data representative of what's really happening, or do I have some aberrant data? And that can be sometimes the real problem. You've got one replicate in one donor that is really throwing things off. And then, I think it's helpful to be able to see, well, what is the impact of that outlier? If you remove that, what kind of an impression do you get? And so I think that's the way that I prefer to think about the role of individual donors versus the across donor population for the sensitivity study.
I just have a follow-up question on the same topic. So when we are doing sensitivity study, let's say that I'm controlling the dose duration. Now, if I have to do the sensitivity study, can I use drug load as a variable or the dose duration? Or which one would I prefer to do it?
So I'd say the first choice would be to use duration. So you're talking about a scenario where this is a wipe-off study, and let's say you've chosen 6 hours as the wipe-off time. So you're essentially modulating the bioavailability by truncating drug delivery after 6 hours. So the first choice would be, to actually then say, well, if I let it go longer, then I expect a higher bioavailability. Now, that's assuming that the flux profile has been characterized and that the absorption phase really continues to, let's say, 12, 18 hours. And so that the product and the nature of the drug delivery is such that left on the skin for longer, you would get higher drug delivery, greater bioavailability up to, let's say, 12, 18 hours. And then you back that off to say, well, I'm going to choose 6 hours as my wipe-off point. And I can show that if my generic product had a different bioavailability, a higher or lower bioavailability, that based upon modulating that's the sensitivity. So for the classic kind of concept of modulating that the sensitivity showing that with higher bioavailability or higher drug delivery and lower drug delivery, that your test would be able to discriminate it if there was in fact higher and lower drug delivery, then that really does a good job of supporting. Once you've got the 6 hour condition and your test and reference product are compared and there's no difference, you've essentially established with that sensitivity paradigm that if there were more or less. Now, if you chose that condition and found some other way, for example, by using a higher strength product and a lower strength product, and that, which it may or may not work, but if you could get that to work and say, hey, it does give me higher bioavailability, that also would provide adequate demonstration that when the drug delivery was different, my method showed it to be different.
Can I add one sentence?
Can we go to Aditya and come back? Because he has been waiting for a long time.
Sure, fine.
Yeah.
Thank you. So just one quick question regarding the selectivity. So based on what I understood, that for establishing the selectivity, is there a requirement for statistical inequivalence or just the qualitative discrimination is sufficient?
Thank you for the question. So as I mentioned in the presentation, the guidance is open for both qualitative and quantitative approach. However, just because of the lower number of donors, it is similar to the IVPT sensitivity. It's very difficult to use the reference scale average bioequivalence because we have seen in some of the application is proposing the reference scale average bioequivalence, which may not be appropriate to evaluate the selectivity. So the current thinking is to evaluate based on the qualitative, similar to the sensitivity study. Thank you.
Thank you.
And I just want to make a note on the previous comment that, I mean, as Dr. Raney mentioned, that it is okay. Sometimes, you can use the consistent or the same dose duration and modify different strategy, like altering the dose amount or product strength. But it may not be appropriate to use the both strategy at the same time. So modulating the dose amount and dose duration together to force the data to be different. So.
Yeah, the only the reason I asked the question is, for example, if I did the dose, the drug load variable as a sensitivity study, would the agency accept it, even though I had used a dose duration? So it's just for people to understand.
Thank you. Go ahead, Theo.
Yeah, for selectivity, we have some kind of flexibility because the guidelines say we can use quantitatively or qualitatively, but when it starts to sensitivity and we use the 4 donors, and there is a question it's a sensitive, it's a judgment, yeah, it's visual observation. And as Allison mentioned, we're getting 70% of the observation because of sensitivity question. And normally, because it's a method development, why not add 4 more donors to more clearly separation? And even guidance says it's a minimum you can do quantitatively or qualitatively, yeah. But you always can do more as a guidance. If you have a sufficient donor numbers, you can run statistical comparison. Why not? This is scientifically proved, and should not be rejected. Also, one more question. Yeah, not the industry clearly doesn't know which product is qualified for a dose duration. Sometimes it's a questions. The FDA, this product is not qualified for dose duration because it's a volatile component it contains, and you have to try. How to try is already tried, and it's not showing.
I think in those cases, the product development meetings or even a control correspondence, if you can
Yeah.
explain the question well, is really helpful, because product specific questions, really looking at your data and understanding why you are proposing what you are proposing really helps us to, for the product development meetings, we have every single discipline available. We have the bioequivalence reviewers, we have the statisticians, we have ORS there. So we can make that decision about your specific program and provide you with our best recommendations.
Correct.
Thank you. Okay, we are going to see if there is some interesting questions online and then switch gears.
For some products that are not petrolatum based, they do not show a decline in permeation and therefore do not have an elimination phase. In that case, what would be the suitable approach to achieve a decline in permeation for a sensitivity study?
Thank you. So I think the question is what to do when we don't have a clear JMAS. Who would like to start that question? Because we have discussed this yesterday. I think Lakshmi would like to go.
Lakshmi, do you want to comment on that?
No, I mean, the guidance is very clear, I think, that if you don't have your Jmax, obviously, first look at the method to see that your method is optimized to get your Cmax and a declining curve after that. So again, I do not know if there is one donor which behaves, where you don't see Jmax, whereas for all the other donors you see, then, again, we do not know if it's considered as a statistical outlier or not. So that debate is still going on, I think. But at this point, my understanding is that your method should be optimized enough to have your Jmax and a declining curve.
Okay, thank you. Thank you for the question. So I'll share my perspective on this one. I mean, we often get this question, how we handle when we have a Jmax at the last sampling time point, and it is connected with what we mentioned in the guidance. So as I mentioned during my presentation, the thought process, to have Jmax and decline in the phase, use the modulation of dose duration approach and things like that for something like a petrolatum based ointment. But I would break it down in a few aspects. I think we heard yesterday how critical, I think from a review standpoint, we would like to see that enough efforts are made to get a complete flux profile. However, we do acknowledge there are certain drug products which do not show decline in the phase. If one would like to use the dose duration or wiping off approach, I would like to make a note that, it can be extremely challenging to remove the applied drug products from the skin surface, and it requires its own method development and optimization. And as long as there are enough efforts are made during the method development, and then you go systematically in your method validation study, both sensitivity and selectivity studies are assessed qualitative in nature. So we are not relying on one endpoint, which is Jmax, and there is a continuous permeation profile comparison between test and reference product. As Sam mentioned yesterday, from donor to donor, you see there is a difference shifting in the Tmax, But taste and reference being a Q1, Q2, Q3 product shows very consistent permeation profile for sensitivity and selectivity. So you have a good amount of data in terms of the sensitivity and selectivity. JMX become more crucial because we are relying on an endpoint for IVPT pivotal study. So my recommendation is, just evaluate the, there is no secondary parameter of Tmax, but if you see comparable permeation profile and there's a Jmax consistent for a taste and reference product at the last time point for few donors, in my personal opinion, it shouldn't be a major concern, but it requires justification in the report.
Rong, did you want to comment also, or?
Yeah, I just want to, I echo what Hiren just mentioned. I just want to emphasize the detailed documentation of, the efforts for optimization during the method development is very important. You need to provide the data to demonstrate a peak flux profile is not feasible despite, various conditions that have been tried. And like Hiren just mentioned, we do look into the individual profiles, individual donor profiles to see if the test product and the reference product perform similarly. So if they perform similarly and also in the profiles in the method validation, the sensitivity study, selectivity study, and also the pivotal study show consistent behavior. I would consider the no attainment of Jmax may not be of concern.
Thank you.
So I agree with all of that. The only thing I would add is we have an obligation with bioequivalence studies to assess bioequivalence based upon the most sensitive and discriminating bioequivalence study that's kind of reasonably possible to develop here. And so when you think about what would push you toward doing a wipe-off study, there may be two factors that would push you in that direction. One is the guidance recommendation that you have a well-defined Jmax and then a decline in the profile thereafter. The focus of that really is more not to push you toward a wipe-off study, but to say, if you've done a 24-hour study, consider a longer study because you may be able to characterize the entire metamorphosis of the test reference product better throughout its metamorphic profile by going a little bit longer. But if you've put reasonable effort into, you've done a 96-hour IVPT study, and it's just the nature of the product that the permeation profile rises, and even in 96 hours, in most donors, your Jmax is actually at the last time point. That is the nature of the product, and I think that's a suitable comparison of test and reference product. That alone should not drive you to doing a wipe-off study. I think you can make good conclusions about the test and reference product there as long as you can show sensitivity. So if you can use, let's say, dose modulation to show sensitivity with those profiles, then do that. If you're not able to show sensitivity and you need to basically go to the dose duration in order to really modulate the drug delivery in a way that shows sensitivity, I think that's more the reason to go toward a wipe-off study, because the process of wiping off, right, which sometimes includes actually just washing it off the surface and a bit of several wipe, that can be more technically challenging. It can make the data more variable. It adds complications into the development, and it can also add complications into the assessment of the data. So I would not go there lightly, just based upon Jmax being, your last profile or your last time point. If you can show during your method development report that it's a sensitive method, it's just that this is the nature of the profile that Jmax is there, I would say that's a good method, and you kind of move forward with that. You don't have to do a wipe-off study. I don't know if you have any further comment on that.
Thank you. I'm going to go to Abhishek for a second for the next question, maybe. So the question, Abhishek, was nominal value versus actual values. I think we heard some discussions about the fact that we want both the values that are available from the equipment manufacturer and actual measurements from our applicants in the application. So the question was, which one should be used for our calculations?
So yeah, definitely, agencies suggest using the actual values in terms of calculating your Jmax or, flux, as that represents the true, true values that you observed. Yeah, I think the actual values probably should be the right one.
Would anybody like to add anything?
Thank you, Abhishek, and I agree with Abhishek. So I just want to make a comment that, as Dr. Raney mentioned yesterday, the old equipment, it was hand blown. And if you look at the cell to cell variability there was a substantial difference between cell to cell. So in those kind of condition, I think it is worth using the actual volume. But if you look at the advancement in the technology and, this new cells some of the cells that I have seen are extremely precise. They are like, plus or minus 2%, 3%, 4%. So it's a very tight window. So ideally, yes, actual value is preferred. But if the variation is 2 to 3% across, across the cell, it may be okay to justify, because eventually you are using those data, you are doing further calculation, converting into using the volume, surface area, and if you use the nominal value, it may be okay as long as it's scientifically justifiable.
Yeah, I agree on that as well, that, like when you qualify your apparatus and the cells, if you make sure that the cell to cell variability is within a certain percentage, you can take it as nominal, but if they are hugely different, then you would go for actuals. And when we have done, we have actually done the same thing, that we got the manufacturer's specification, but we also did our own qualifications of the cell. And we normally keep the same cell in the same position in the console. So, we know which cell is which, so we can always evaluate as we go along in the project.
Thank you.
Yeah, we have annual qualification procedure is plus minus 5%. And if cells not complying with this one, is disqualified. So it's calibrate every year, so 7 years. And so if you don't use the nominal value, it's a lots of work for the document reviewer, and they create unneat works.
Thank you.
I think the only time there's a challenge is when your cell breaks and then you are replacing. You want to make sure that that cell is within the same range as well. Otherwise, you have to make sure of how you are using that cell volume in the calculations.
Yeah, I just want to make one comment, as Dr. Schafer mentioned that the expectation is not to qualify for each and every study, as long as you have a system in place for the lab that you are qualifying this equipment every 6 months or a year, and there should be reuse in multiple applications, and it should be sufficient. Thank you.
Okay. So in the last maybe couple of minutes, outliers. We have heard about them yesterday, we have heard about them today, and we are going to discuss them in our final panel as well. But a more maybe high-level question about outliers, methodology related. We have seen some examples where tune measurements are proposed not only at the beginning of the experiment, but also during and after the experiment, and using those transepidermal water loss values to justify exclusion of data from the pivotal study data set. So I would like to get your thoughts on, one, what does a measurement of TWL at the end of the study tell us, and can those be used to exclude cells?
Just in the interest of time, I'm going to cut to the chase here. I find it very difficult to interpret TWL results from a cell that's already been dosed with the product, because it's very difficult to know what the impact of the product was on the diffusion cell. Perhaps that's a feature of the design of the product that it alters the barrier. So I think the real value of TWL is as a barrier integrity test, as an exclusion criterion. I don't know that, I don't personally know how to interpret those results after the cell has been dosed. But I can understand why someone would say, look, this is supporting evidence for a barrier function anomaly. But I just don't know that we know enough about the relationship between that TWL result, the way the formulation affected the skin, and the barrier function that resulted to really connect the three of them, at least not for me.
Any other thoughts on that?
Normally, when you're running the pivotal runs and the pilot runs, we always included non-dosage donors. So we can monitor during the run each time point how the donors' behavior, monitor the TW. This can give you the ideas if these donors during the study duration is still okay.
But Theo, a follow-up question then. Does your non-dose cell give you similar TWL values as your dose cells throughout the duration of the experiment?
It can be varied, but it's still in the range. Acceptance range.
Thank you.
I also just want to add a comment. I agree with what Sam said. Like, I think also probably the overhydration over the duration of the study will also skew the results for the TWL if you take that into consideration for identifying that as an outlier. So sometimes that gets tricky as well.
Thank you. Okay. Any final thoughts? Go ahead.
I do think it's worth having more research in that area. Yeah.
I have another question regarding any product which has very low strength, very, very low strength. In that case, it is sometimes almost difficult to find the permeation of the molecule in the receptor solution. When we apply our target, like maximum 15 milligram per centimeter square, then what could be our approach? Increasing the dose application or what else? How we can go ahead? Thank you.
Would anybody like to take that question?
So after the exercise yesterday afternoon, you should all be able to help with that question.
Fair enough.
But I think in the interest of time, Priyanka, I'll let you decide how you want to handle that.
I think we can discuss offline. I think this is definitely sounds like a product specific question. So I would encourage you to, discuss with us through the product development meetings or controls when appropriate. But like we discussed yesterday during the session, analytical sensitivity, we try to make sure that the study is actually doable before we recommend it in a product specific guidance. But we are aware there are some challenges with some of the products, such as, ivermectin, tacrolimus, we talked about it yesterday. So if you are finding those challenges, I would encourage you to come and talk to us. But like Sam said, in the interest of time, we actually want to give you a coffee break today. So we will return promptly at 10:40 to continue with the discussions. Thank you so much. Thank you.